Hi, I'm Helena Highsong, and I'm going to be talking to you today about a undergraduate research experience that I had within a course, which was the genetics lab. So we were looking at the adaptive osmotic stress response of sourdough starter fungi. So what are sourdough starters? Sourdough starters are basically just a mixture of flour and water. And what makes them special is they're going to be fermented by any wild yeast and bacteria that kind of just find their way in there. So each one's going to be unique to its environment based on what organisms from that area find themselves in the starter. So how does this work? The yeast will break down the starches in the flour, and then they will produce carbon dioxide as a byproduct of cellular respiration. And this carbon dioxide will be seen as these little bubbles in the starter, and they make the starter rise. So what were our goals for this research? We wanted to attribute our phenotypic data to genotypic data. So our phenotypic data was going to be the rising rate. We were going to test out different starters and see how quickly they rose under various conditions. And then we wanted to attribute that to the osmotic stress response. And we were going to do that to answer which microbes in our starter communities and how did they respond to osmotic stress. And then over time, if grown in a salt environment, can those microbes adapt to the salt stress? So basically, will starters that have been grown in the salts handle salt stress a little better? Is it selecting for microbes that have a better osmotic stress response? Methods, how did we do this? We had a class of 178 students. There were in-person students and online students. So the in-person students actually measured the sourdough starter rising rates at the various concentrations and did ITS PCR, which I will explain in a little bit what ITS is. And they did that to identify which fungi species were in the different colonies in the salt starters. And then meanwhile, the online students did shotgun metagenomics, which is basically looking at all the different types of organisms in the starters to determine the microbial makeup of the various starters. So what is ITS sequencing? It's a type of DNA sequencing, which determines the nucleic acid sequence that make up each organism. And ITS is an internal transcribed spacer in fungi. So it's a barcode region. What this means is almost all fungi have this region so that we can design primers to make sure we're getting the ITS of every single fungi, but each species has a different variable region. So then we can determine which fungi and different species of fungi are in our starter. So how did we start this experiment, our control starter experiments? This is actually my rising rate graph. I was an in-person student. So you can see that we have our control, and my sourdough starter was grown in 0.25 molar sodium chloride. And for some reason, we're still not quite sure. The starters in the 0.25 sodium chloride actually had a better rising rate, which was not what we were expecting because typically salt is a stress which would slow down the success and the rising rate of the communities. And then we took all the course averages from all the different classes and students using different concentrations, and we were able to make this graph. And what this graph shows us is that consistent with my data, the 0.125 and 0.25 molar sodium actually for some reason grew quicker than the control. But you can start to see that at 1 molar salt is when the sourdough starters actually start to decrease in rising rate. And then 1.5 more significantly so, and then around 2 and 4 molar, it pretty much completely kills the communities. So here we actually have our streak plates on the left. We took different species from our starter and we made these different colonies. And then we put these on a gel to PCR the different colonies. So as you can see in the first lane is our ladder, and then we have a control, and then you see very faded bands for our samples. So my actual data was not able to get good PCR results because our bands are very faded. It just wasn't good experimental procedure. But Luckily, we had the entire class, and there were some groups that were able to get some data from their ITS region and identify the different fungi. So the main fungi found across all the different groups was this Kazakhstani humilius, which is a pathogen, but it's a common yeast found in sourdough starters. And then we had a couple of others down here. We had a couple of Pichia genus fungi, and this cross here between Saccharomyces cerevisiae, which is common in beer, and Bianus. So then the next week we have our rising rate graph 2, and this time we used 0.5. We still use our 0.5 molar NaCl, and now we can compare the control with water, which is just a sourdough starter that hasn't been exposed to any salt stress, and then a similar control that hasn't been exposed up until this point to stress, and then we're going to expose it to that stress. And then we also have our control, which we have been growing in the salt. And then what happens when we take our control that's been growing in the salt and expose it to more salt? So the idea is, since it's been growing in the salt, it will have adapted to osmotic stress, and it should perform a little better than this control here that's never seen this salt stress before. And as you can see, that is the case in this experiment, but I wouldn't exactly say significantly so. They're all still pretty similar rising rates. And then once again, we had a bunch of course averages for all the different groups that did this experiment. And where there started to be a significant difference was at that 1 molar concentration. So as you can see, the control exposed to salt, so the sourdough starter that's never seen salt before, it rises at this rate, while our control that has been exposed to salt before rises at this rate. So once again, we're actually seeing a drop, which goes a little bit against what we were thinking. And then we have our, once again, we did the PCR, and we have our control of the control, and then we have different species that it's the same thing with the colonies, where we had different colonies from the control, and we ran these here. And then we also had our samples, which had been exposed to the salt, and we ran those in lanes 9 through 12. And as you can see, we actually do have 3 good bright bands here in lanes 2, 5, and 9. So those were actually successfully used in PCR this time. So once again, our class all had their different PCR results, and ours were included this time. And once again, the major fungi found was the same Kazakhstani humilius, which is a common yeast found in sourdough starter, so no surprise there. And we also had this cross here with the Saccharomyces again. And then we had this new pathogens that we weren't quite expecting, that we think is from some sort of contamination. Okay, so once again, now we're going to do this same experiment again, but this time we used a stronger salt concentration because the lower ones weren't quite doing what we expected. But as you can see here, there is actually a bigger difference this time since we used that higher salt concentration. So the control is up here. The control was salt. And then also down here is what we had been growing in the salt, and then after it's been exposed to the salt. So, and then once again, we have the class average, and it was also the 1 molar where we started to see that difference. So when comparing the fungi composition in our control versus when we have been exposing it to salt, our Pichia genus fungi actually disappeared, which likely means they're not quite as good with the salt stress response, and it perhaps killed them all off, even though other species were able to still stay alive. So for future directions, we would probably want to incubate these in the salt for a little bit longer, just to see a bigger difference, allow them more time to adapt to that stress response, more generation times to select for species that have better stress responses to this osmotic stress. And then as you can see from our very first experiment, actually the amount of salt that started to kill off those species was at about 1.5 molar. So this would probably be the best salt to incubate our sourdough starters in, because it is killing off some species, but still some are still staying alive. So those are probably the ones with the better stress response. And those are the ones we'd want to select for and see if overall we can get that adaptive response. So luckily in future courses, the students are going to redo this experiment, keeping our findings in mind. So it should be enhanced, and hopefully they'll get a bit of a better result than we did.